Sarah is a professor here at UBC in the Faculty of Pharmaceutical Sciences. She's going to speak to us today about topical gene delivery. Dr. Hedtrich obtained her PhD in pharmacology and toxicology from Freie Universität in Berlin in 2009. During her postdoc, she moved to Ludwig Maximilians University in Munich and Tufts University in Boston. After returning to Berlin in 2013, she headed a junior research group and was appointed an assistant professor in 2015. She relocated her lab to the University of British Columbia in 2019. Her research focuses on inflammatory and genetic diseases of human epithelia, the development of novel therapeutic approaches, and tissue engineering of complex human-based organ models aiming for valid and predictive tests for preclinical and fundamental research.
So thank you very much for inviting me and for giving me the opportunity to present our work. And I should say that we just kind of started in the really gene therapy and topical gene delivery. And so what I will try to do in the next few minutes is to give you an idea about the challenges when it comes to gene delivery, especially when we talk about local gene delivery. And our focus will be especially on the skin and the human lung, basically. So what I'm planning to cover is I will talk about delivery to the skin in general, because this is a major epithelial barrier. And I will talk about what we did over the past 6 to 7 years, and I will introduce the disease we are mostly interested in. And then I will give you a very first data from our lab, where we just started actually the CRISPR delivery into the skin and into the skin cells. And in the last part, I will just briefly comment on topical gene delivery to the lung and the challenges which come when we talk about CRISPR delivery to the lung. So when we talk about the human skin, you should know that this is the biggest organ of the human body. And it looks quite simple, but actually the skin is a very, very complex structure. And it is composed of several different layers, starting with the subcutaneous fat layer, then the dermis layer, and the outermost layer, the epidermis. And the epidermis is really a very, very thin layer. So in humans, it's between 10 to 30 micrometer. However, it is a significant barrier. And the skin offers several advantages when it comes to drug delivery. So it's very easy to access, so we can just simply, that's what we are all doing on a daily basis. We are putting creams onto our skin and we hope that we don't get wrinkles or that it treats our skin disease, so that's really easy. We can apply it locally, and this is actually something which is really favored by patients, so that you don't need to go systemically. Furthermore, when we apply drugs to the skin, we can avoid first-pass effects, which mean that we can go down with the drug doses, because if we administer something systemically, so if we swallow a pill or if we inject something, it directly goes to the liver and it's instantly metabolized, and then the actual drug dosage, which reads the target organ, is much lower than what we initially administered. And with the skin, it's much easier, because when you put it on the skin, although there are some enzymes, but it's not that extensive, and so you get most of the drug which you apply to the skin, then finally also reaches the bloodstream. And you also have reduced side effects, especially when you are targeting the skin. So, there are some clinical some diseases like psoriasis, which also affect the entire body, but there are also skin diseases, for example, like atopic dermatitis, or the one I will talk about in more detail, which almost exclusively affect the skin, and so a local application of the drug, or in this case of the gene therapy, is clearly of advantage. So it seems like the ideal way to go, so a topical application like a cream onto the skin. However, there's a big but to that. So I already mentioned that the skin is a very, very restrictive barrier. And with very restrictive, I mean that only very, very few substances can readily penetrate the skin. And those substances should normally be very small. And with small, I mean a molecular weight below 500 Dalton. So not kilodalton, Dalton. So this is really, really small. And they should have a moderately lipophilic log P between 1 and 3. And so when we talk about CRISPR and gene delivery, you can already imagine that it's kind of tricky to deliver this efficiently into the skin, because it's way too big. So we are in kilodalton, and so this is really huge. And so in the past, I think so about 10 years ago, there was the big opinion that you can never deliver biomacromolecules to the skin efficiently. And so about 7-8 years ago, we became interested into a rare monogenic skin disease. And this rare monogenic skin disease is called autosomal recessive congenital ichthyosis. The abbreviation is ARCI. And basically what happens is that those patients develop very, very scaly skin. And I should say that this is a very nice picture. So if you Google this disease, there are really, really bad phenotypes of this disease. And so the thing is that there are different phenotypes. And some of them, they are associated even today with a mortality rate of about 50%. And the reason for that is because those children are born with, or in general, those patients have a severely impaired skin barrier function. And especially as a newborn, they have a massive transepidermal water loss, and they are prone to skin infections. And so they need to be kept in a special incubator. And again, as I mentioned, still survival rate is 50%, especially of the children suffering from harlequin ichthyosis. And so it's a rare monogenic skin disease, and there are 11 causal mutations known so far. And the most common mutation occurs in a gene which codes for the enzyme transglutaminase 1. So up to 60-70% of the patients have a mutation in this particular gene. And what this gene codes for is an enzyme which is crucial for the cross-linking of the outermost layer of the skin, the stratum corneum. And if the enzyme is not expressed, then there's no cross-linking, and this is the reason why you have this severely impaired skin barrier function. And I should mention that there are no treatment options at all. So the patients need to take oily baths on a daily basis, ideally twice or thrice per day. They need to apply very, very fatty creams or very rich creams several times per day. So it has a very, very significant impact on their daily life. And so back then what we thought was we know exactly what's wrong with the patients. We know exactly which protein is missing, and in this case it's transglutaminase 1. And why can't we just simply replace this protein? And I'm telling you this story because this is kind of the first steps toward the gene therapy we are currently working on. And so the protein is like 90 kilodalton big. And we knew about the problem and the perception that macromolecules cannot penetrate into the skin. But we thought, okay, the skin barrier function is severely damaged, so we thought there may be a chance that we can actually reach the barrier. And I already mentioned this. The problem is that this is normally possible, and we need to deliver this. So this is really big. And then we had another problem, because for this ARCI disease, there are no animal models available. Because what happens is you can generate knockout models, but the animals die immediately after being born. And there have been some transgenic, some models where you transplant the skin onto a mice, and then you can overcome the lethality of the disease. However, the problem is that the skin and the disease behave differently in those mice. So what we thought about, okay, animal models are not available, and the other thing you need to keep in mind are the interspecies related differences. That you see the difference between mouse skin and human skin. If someone works on mouse skin, it's way easier to deliver something into mouse skin than in human skin. And I mean, all the data are important and it's very nice, but if someone says they can deliver CRISPR here, that's way easier because mice only have like 2 epidermal layers, whereas in human skin you have like up to 20 epidermal layers. So that's a completely different story. And there are a few other things like the mice have fur, we don't, there's another cutaneous layer, and so on and so on. And there are also immunological differences and differences with regard to the regenerative potential. And then also when we think about the gene therapy, there's only a 30% overlap in skin-associated genes between man and mice. And so for us it was clear that we would need like a human-based model to study this. And what we do is we do tissue engineering of those skin disease models. So we can reconstruct full thickness human skin by using primary human cells. And to model the disease, we can either knock out the genes of interest in the keratinocytes, or we can generate patient models. So we, back in Europe, we actually recruited some ARCI patients. That's very tedious work, so we finally ended up with 2 patients. So we got skin biopsies from them, and we are also able to isolate the cells from plucked scalp hair. And those cells, they retain the genetic information. We can expand them, and then we grow our skin models which emulate characteristics of this disease. And this was basically then our model which we were using. So coming back to the delivery of the protein, because we wanted to prove that it's possible to deliver biomacromolecules into the skin. And so we teamed up with a polymer chemist, he's now in Spain, and together with them we developed thermoresponsive polyglycerol-based nanogels. And basically, without going into too much detail, what those nanogels do is that they can efficiently incorporate proteins, and that they can undergo a reversible phase transition at a certain temperature. And when at this temperature the nanogels shrink, and due to the shrinkage, the loaded proteins got expelled. And so our idea was that we know that there is a natural thermal gradient of the human skin. So on the skin surface, you have a temperature of 32 degrees Celsius. But as soon as you reach the viable layers in the viable epidermis, you are around 37, and somewhere in between, there is a turning point of around 35 degrees Celsius. And so we designed the nanogels that way, that at a temperature of 35 degrees Celsius, they would undergo this phase transition, they would shrink, and then the protein would be expelled. And we hope that this would be sufficient to facilitate the delivery of the loaded biomacromolecules into the viable layers of the skin, which is our target tissue. And so when we talk about protein encapsulation, everybody is very concerned about the stability of the proteins, because these are very labile substances. And so we did a lot of stability studies. We looked at the structure of the protein before and after encapsulation, which worked quite nicely. We also looked at the loading, so after loading, how stable is the protein after storage or during storage, and so we kept it for several weeks. And it seems that as soon as the protein is loaded onto the nanogels, the nanogel kind of protects the protein as well, rather than it's in solution. And another critical point for us was also the release of the protein from the nanogels, because this needs to happen very fast. And so we did some release profile on that, and for us the critical point was that the release really happens at temperatures above 35 degrees Celsius. So if that would happen at 32 degrees Celsius, which is on skin surface, the protein would just lay there and nothing would happen. And so we did, I think the caption is missing here, but basically these are different temperatures. So this is room temperature, this is 32 degrees Celsius, and this is 37 degrees Celsius. So we were able to show that really that our approach and our principle works, so that the protein really gets expelled at higher temperatures. And then in the next step, we need to provide proof of concept that we can actually deliver it in human skin. And so we used our skin disease models, and we used 2 different ones. So one was a model where we artificially induced the gene knockdown, and the second model was a patient model. So we, as I said, we were able to recruit some ARCI patients for this study and generated then the disease models with the patient cells. And then we applied our transglutaminase-loaded nanogels topically, and the very first experiment was to see whether the nanogels are indeed able to deliver the protein into the viable layers. And what I hope you can see it here. So this is a cross-section of a normal skin model, and you can see in red the expression of the transglutaminase-1 in the viable layers. And this is a disease model, and I hope you can see that here the transglutaminase-1 expression is significantly reduced. And after treatment with our protein-loaded nanogels, I hope again that you can see that we have now, again, a very bright red signal in the viable epidermis, which indicates that indeed the protein has been delivered into the viable epidermis, which gave us the first hint that the delivery of biomacromolecules is possible into the skin. But again, we are dealing with a severely damaged barrier, so the chances were good. Let's put it this way. We then had to provide, we had to prove that the protein was not only there, but that it's still functional. And so we did an in situ activity assay of the transglutaminase 1 activity. And what you can see here is that this is a cross-section again of a normal skin model, and we see that we have a nice green signal, which means transglutaminase expression is there and it's also active. And this is now a patient model, and as expected, there's no enzyme activity at all. However, after treatment of the patient models with this enzyme loaded onto the nanogels, we again see that there is an enzyme activity in the viable layers, and this is also where we need it. And so in the end, what we needed to provide is if we are able to, or to prove is if we are able to rescue the barrier, because it's severely impaired, and our aim is to rescue the barrier function and to improve the barrier function. And so what we did were 2 sets of experiments. So the first one is, this is basically the control experiment. So we did some permeability assays so we can determine the barrier function of the skin models using some radioactively labeled compounds. And what we did was we applied our therapeutic protein in a simple buffer solution topically. And what we see is here in black, this is the permeability of a normal skin model. And in gray, you can see the permeability of a patient model. And then we applied our therapeutic protein in increasing concentrations in a buffer onto the patient models. And what you can see is that there's no effect at all, so nothing happens on the skin barrier. So which is good, which was expected. And then in the second step, what we did, basically the same setup, but now we applied the therapeutic protein when it was encapsulated into the nanogels. And what you can see now is that we see a nice dose-dependent decline of the permeability in the skin models. And this, in the end, was the proof of concept that the protein is not only there, but that it's functional, and that then it can really do what we were hoping it would be doing, and namely rescuing the severely impaired skin barrier function. And so these studies kind of laid the basis for the gene therapy we are currently working on. And with that, I'm now coming to the gene therapy of monogenic skin diseases. And when we think about that, before I come to our recent data, I just want to introduce you a very nice study which was published, I think, last year or the year before, on another rare monogenic skin disease, which is epidermolysis bullosa. And this is, again, a very heterogenic and inherited skin disease, and several genes can be affected. And here the problem is that the patients are experiencing a severe blistering. And again, this can be a life-threatening disease. And similar to ARCI, this is a very good example, or a very good a disease which could be easily, or hopefully easily, addressed with CRISPR-Cas. And so what the patient, or what the clinicians did in this case was they applied, or they used CRISPR-Cas to rescue the phenotype of an 8-year-old boy. And you can see the boy here. And actually, it was only approved because, he had a very, very severe type of this disease. And this was also almost lethal condition. So it was like the last resort for this kid. And what they did was they took a skin biopsy from a healthy piece of skin of the body, and then they expanded the cells from the skin biopsy. And then they applied CRISPR-Cas to those cells, and they used a retroviral vector in this case, and the gene was restored through CRISPR-Cas. And what they did then was they expanded the cells massively, and they grew a very, very large piece of skin, and they retransplanted this skin onto the body of this boy. And what happened was basically that this saved his life. So the skin really grew onto the body of this, so the transplant worked, the transplantation worked, the skin really connected with the body of the boy, and so this is what it looks like after, and there was almost no blistering anymore. However, As I already mentioned, it was done with a retroviral vector, and there are several safety concerns associated with those retroviral vectors. And so this leads me to the project we are currently working on, which is the development of an in situ gene therapy for rare monogenic skin diseases. And our approach is a bit different to what the clinicians did in the study I just presented to you, because what we want to do is a local application of CRISPR-Cas. So no skin biopsies and then expansion, but we want to apply it ideally like a cream at some point onto the body, and to deliver the CRISPR-Cas into the skin. And for this, we are closely working, especially at the moment, with the Cullis lab, and we are using lipid nanoparticles to deliver CRISPR-Cas into the skin cells. And we are trying also other particles, so polymeric nanoparticles, for example, also lipoplexes. And we are trying to deliver RNPs, but also the mRNA, and we are just trying to find the best setup for the local delivery. And I should say that skin cells, and especially primary keratinocytes, are especially difficult to transfect, and I can show you an example in a few minutes. And the idea is that we probably, in addition to the delivery systems we need, so like LNPs, we probably need another technique, because most of the nanoparticles which we apply to the skin, and I've been applying a lot of nanoparticles to the skin, so this has been, I dedicated a lot of my time and my career to topical drug delivery, and all of the particles we applied to the skin, most of them really facilitate the drug delivery, but 99% of the nanoparticles stay in the stratum corneum, so they cannot overcome it. And even if in the severely barrier damaged skin, so like in the archy skin models, we never saw that more than 5% of the particles actually reach the viable epidermis themselves. All they do is facilitate the delivery, which is good in a sense because then it reduces the toxicological concerns. On the other hand, for CRISPR-Cas9 delivery, we need the nanoparticles to really reach the cells in order to transfect them. And so we are thinking we may need to use different techniques. So we are thinking about applying microneedles, iontophoresis. There's also some suggestions about IV injection, so that you inject it intravenously and then it gets distributed. I'm not really sure if that would work, but this is just a collection of things which could be done, and we are trying to explore that. And so what we are currently doing, and to be honest, it takes us much longer than I initially expected, is to really find suitable delivery systems to transfect the keratinocytes efficiently. And as I just mentioned, we collaborate with Peter Cullis in this regard, and we tested some of his lipid nanoparticles. And at the very beginning, I met with his postdocs, and we just said, okay, let's give it a shot. Let's go for the most common LNP formulation. And we did that, and in the end, we ended with a transfection efficacy of 13% in skin, so in the primary keratinocytes, which is not really a lot. And when we talk about archy, we need to keep in mind that this is a recessive disease, which means that we also need to provide a donor template. So we need to do HDR. And in the end, the efficacy of HDR is much lower than if you just need like a knockout. And so if we only can get a cutting in 30% of the cells, the HDR rate, so the correction of the gene, that's, close to zero probably. And so what we decided is to really take a step back and to do a more thorough screening and more comprehensive screening. And just to give you an idea of what we currently do and how different the results are, is that we are currently screening really a broad library of LNPs, different helper lipids, different concentrations, different PEG concentrations, different helpers like ApoE or no ApoE. And you can already see from this bar that there are significant differences between the lipid formulations we are using and testing, but also that already if there's ApoE or no ApoE, or if there's 0.5% PEG or 1.5% PEG, we also tested 5% PEG. So there are huge differences. And so we'll probably need to spend a few more months on that to really identify the most efficient delivery systems. And So yeah, this is definitely necessary. And what I not mentioned yet is that when we think about a curative approach for ARCI, we probably need to target the skin stem cells, which are in the basal layer of the skin. And the skin stem cells, they are considered like the holy grail. And we run some studies on transfecting the skin stem cells, and this is even more difficult than transfecting efficiently the primary keratinocytes. So I guess that there's still a lot of work to do. And in the last few minutes, I just want to briefly talk about the lung delivery, because this is another human epithelia. And so when it comes to lung delivery, we need to approach this from a completely different angle. Because the lung, although it's also a human epithelia, there's no stratum corneum, but there are other barriers, normal barriers. And the big issue with the lung and also the gut, which is another epithelia, is basically that you have the expression of a thick mucus layer. And when we now talk about delivery to the skin, and then we talk about delivery to the lung, we may need completely different delivery systems, because in order to overcome the mucus of the lung, you need completely different particle properties than when you want to overcome the human skin. But nevertheless, I truly believe that CRISPR and the local delivery of CRISPR-Cas is especially promising for a very common lung disease, which is cystic fibrosis. I'm sure you've heard of that. That's the most common fatal genetic disease in Canadian children and young adults. And again, there is no cure. There are different severities, and it not only affects the lung but also the digestive system. But most of the patients that die from cystic fibrosis die because of complications in the lung. And here it's pretty similar to the ARCI disease, which I just talked about. There's one very common mutation, which is this mutation, and this mutation occurs in 70% of the cystic fibrosis patients. And so this is again also a recessive disease, so you would also need the HDR approach, so not just the knockout approach. And just to give you an idea of what you need to consider when we want to deliver CRISPR-Cas to the lung, is that there are different strategies for the nanoparticles to deliver things through the mucus and to finally reach the epithelial cells, because these are the cells you need to edit in the end. And there are several approaches, like either using mucus-penetrating particles or mucoadhesive particles, and there has been a lot of work done in this regard, and it seems that nanoparticles with some mucoadhesive properties, but if they are still able to slip through the mucus network, that these are probably the most efficient delivery systems when it comes to lung delivery. And in preparation for this presentation, I also looked up, or I looked at PubMed and tried to find some local deliveries to the lung of CRISPR-Cas. And actually, I just found one, so at least published. I know there are some people working on that, but I just found one so far. And this has nothing to do with LNPs. What they did here, they used Shuttle peptides. And so these are different peptides which have been designed, and those peptides, this is a combination of cell-penetrating peptides and endosomolytic peptides, and these were used as delivery systems for CRISPR-Cas to the lung. And what they did, they tested the editing efficiency in mice in large and small airway regions, and just that you see that here the green dots, the green dots indicate gene-edited cells in the lung of mice. But again, how representative this is for the human situation is questionable, because the anatomical layout of the mice is different, the mucus composition is different, and so I think there's still a lot of room for improvement, and there's a huge need for efficient delivery systems for the lung, and also the assessment of this. So what are the ideal systems, especially when it comes to CRISPR-Cas and gene delivery? So with that, I'm at the end. I would like to thank the people that contributed, the funding agencies that gave me money for my research. And I would like to thank you, and I'm happy to take your questions.
Questions for Cynthia? A very good talk. And in your lung delivery part, you mentioned that the 70% of the patient who has the CFTR mutations has the disease. And may I ask, what's the effect after the lung delivery and modify the deletions?
I'm not, can you repeat?
So my question is that for this kind of 70% patient who has the F58 deletions, right? Once you deliver your nanoparticles to this kind of patient, what's the effect of this delivery in this patient?
And to be honest, I don't know. So the idea would be that when you deliver CRISPR to the cells, to the basal cells of the lung epithelium, that then the mutation would be corrected, and then there would be an alleviation of the symptoms, basically. So that is the idea, but it was just an example for a disease where CRISPR could be useful in the future.
Okay, so for the treatment effect, you didn't have the data yet, right?
No.
Oh, thank you.
You were talking about the thermal gradient across the stratum corneum. And I just wondered if there was any way of exploiting the pH gradient that's also, at least in instances of healthy barrier, present.
Yeah. So that's a good point. That's actually a very good point. The thing with the pH gradient is that healthy skin has a pH of 5.5 on the surface, and the deeper you go, it gets neutral. And from a particle design perspective, it's very difficult to go into this direction. So the other direction would be easier, so to design particles which would degrade if it goes the other way around. But so we thought about that as well. But the chemistry behind that is way more difficult than the other way around. And that's why we went for the temperature gradient.
Have you looked at using photonics or acoustic ways of temporarily kind of increasing the porosity of skin to help to facilitate delivery?
That's a very good point. So we thought about it. We are currently so that would be something on the to-do list. We haven't done it yet. We are currently working with the microneedles, and we are exploring the iontophoresis approach. But we are open to everything, and that's definitely something we can look into. Yes.
Do you think it's feasible to combine the nanogel and LNP techniques? So is that maybe something, if you have an optimized LNP formulation in vitro, do you think you'll still need a way to get it past the barrier? Do you think something like a nanogel approach will work?
So the nanogels themselves, they do not overcome the stratum corneum. So they stay in the stratum corneum. They only facilitate the delivery of the biomicromolecule. And again, this is nothing what we want or what we need for the CRISPR delivery. So we need particles or transfection agents for the keratinocytes themselves. So we need to find a way to really deliver the particles as they are to the cells. Initially we thought about the nanogels, but they cannot encapsulate CRISPR. So we are currently redesigning them. So we are trying to make it work, just to test different angles. But what I can also say, and this is well in line what the previous speaker mentioned, is that we indeed also observed the uptake of not encapsulated RNP into the cells, which was unexpected, actually. And it's very similar to what he mentioned. So we never thought about that, and we put it aside and said that must be a mistake, but you mentioned it as well, and we see similar things in the keratinocytes. So maybe we shouldn't put it aside, maybe we should think about it a little bit more.
Yeah, you were getting only 13% transfection of the keratinocytes. That's with the straight non-factor formulation. But with those, I wasn't quite able to see from your slide with different lipid compositions. Were you doing appreciably better, or was it?
So what we took several steps back. So I cannot answer this question yet, because what we currently do is we try to identify the LNP which is most effectively taken up by the keratinocytes. So currently we did not yet look at the cutting efficiency. So we just deliver some, I think it's fluorescently labeled as iRNA, to the keratinocytes to see which particle can most efficiently deliver it. And in the next step, so we already narrowed down the particles which seem favorable for this approach, and the next step will be to reevaluate the cutting efficiency, and we hope that we will get better results this time.
Any further questions?
Maybe just a comment also related to the question a person in the front asked after the nanocore introduction. Sarah's study is really a prime example for the optimization we try to do with the different investigators. Like Sarah has shown, like on Petro-like formulation is optimized for siRNA for liver gene therapy, but it's maybe not optimized for skin and RNP delivery. So we try to then screen different formulation, optimize it for the different nucleic acid payload and for the specific tissue. So yeah, as you mentioned, we try to find now which one shows the highest uptake, and then go a step forward and see which this translates also to a gene therapy effect.
Anybody else? Okay, well, thank you, Sarah, very much. That's great.